All right. Hi, everybody. Can you hear me okay? I have this mic on. I don't know. I have a pretty quiet voice, so okay. Awesome. Well, thanks for having me. Today, I'm going to be talking with you about some of the work that Atlanta Botanical Garden is doing down here in Florida. Just to give you a brief overview of what we have in store. First, I'm going to give just a little brief background about ABG, and then I'm going to highlight our work by zooming in on our work with Rhododendron Chapmanii. And at the end, I think we're going to have a very short amount of time to talk about two other projects, just enough to hopefully give you some questions. If you want to learn more about those projects, you can come up to me afterward. But we're mostly going to be spending our time with Chapman's rhododendron. So when people think about Atlanta Botanical Garden, I think probably the first thing that comes to mind is the physical space, the landscaping, a place where people can go and build a relationship with nature in an urban environment. But we also have a whole team of researchers working on plant conservation. And that's happening within the Southeastern Center for Conservation, of which I'm a part. That's where our conservation and research department is. And our focus is primarily in the Southeastern United States, but we also do work in Ecuador and in Puerto Rico. And we're also part of international consortia for specific groups of plants. One of those is Magnolia and another is oaks. Our team is down here in the bottom left at a strategic planning retreat. Our Atlanta-based team primarily works in the Southern Appalachians and in the Piedmont of Georgia, but myself and a couple others work primarily and only actually in Florida and Alabama. So we approach our plant conservation work in a couple of different ways. Because we have a physical garden space, we have a team of conservation horticulturists who work on growing out plants outside of their wild habitats, so ex situ conservation. We also have a seed bank and we have a genetics biorepository. And so those are ways where we can conserve species and preserve genetic lineages outside of their natural habitats. But then we're also doing work in wild habitats, because for us, we don't want to just conserve a plant just for the sake of its existence. We really want to keep plants relevant and do what we can to learn more about the ecosystems in which they occur and do all that we can to keep them relevant. Down here, I have a few numbers. So our conservation horticulture team is headed up by John Evans. He is a wonderful conservationist, and he's currently working on growing over 10,000 plants from 300 different species. A lot of these end up back in wild settings, either as reintroductions or as augmentations. And then our seed bank currently has over almost 3,000 accessions from over 500 different taxa. And then our biorepository has almost 9,000 different accessions. And they're available for other researchers who are working on similar species for us. So let's get to the Chapman's rhododendron. I'm really excited about this plant, and I think it's a great way to highlight our work because we're really using all of the different approaches that I mentioned, the ex situ and in situ methods to try to keep this plant around. But before we dive specifically into this, I wanted to go into just a little bit of backgrounds about rhododendrons in general. So the map at the whole left pane comes from Jamais et al.'s 2021 paper that was looking at reasoning behind rhododendron speciation. But the top pane shows you the distribution of rhododendrons globally. And the most biodiverse areas are in the Himalayas and in the Southeast and Southeastern Asia. This paper looked at why that is and found that areas that have high precipitation and also a wide range of elevation available really leads to the rhododendron genus branching out into more species. So it's a really important plant. There are 1,000 species of rhododendron, and where they grow, they're key components of their ecosystems. They're shrubs, and so they perform a lot of structural roles like providing habitat and shading streams to keep the fish able to exist in those streams. So they're incredibly, incredibly important. They're also really important to the humans that live and exist with these different species. So I have a couple examples here. The top two pictures are of rhododendron maximum from the Southern Appalachians. If you go up there, it's very common to find people building with rhododendron, building beautiful structures, carving bowls, spoons. And then in Asia, especially, rhododendron wood is used for warming homes, and the flowers are really important in spiritual ceremonies. Rhododendron are toxic, but they've been used by herbalists around the world in medicinally. So I just have a couple examples here. There's a species in the Himalayas that is used in a wine to help with altitude sickness. And there's another one that's also in the Himalayas that is used for lung issues. And so it's made into incense, and it's used, again, ceremonially as an essential oil. So these plants have long histories, and here too in North America, native tribes have long used them. Again, rhododendron maximum comes up, and calendulaceum, as well as some species from up in Canada that are used to prevent diabetes. So these are critical plants for ecosystems and for people who are also part of ecosystems. In North America, the pollinators of rhododendrons tend to be butterflies, moths, bees, but in Asia, it's birds. So I myself do not know who each of these birds are, but they are the important pollinators in Asia primarily. So coming back to North America, there are about 30 species of rhododendron. And in Florida, depending on who you ask, there are either 5 or 6. So Weekly's flora breaks the rhododendron viscosum out into rhododendron cerulatum as well. But the Florida Plant Atlas considers cerulatum to be part of the viscosum complex. These plants tend to be in wetter areas, and they do go all the way down pretty far down into the peninsula, but most of these species are found in the northern part. It's rhododendron viscosum. I believe that's the one found further down into the peninsula. But today, we're going to be talking about rhododendron chapmanii, which is a federally endangered species. There are 2 other state endangered ones, which are alabamense and ostrinum. But here we have Chapman's rhododendron. So they have pink flowers. They have leaves which are pretty thick and rolled over. And the leaves are also marked by a characteristic I did not get a good picture of. They are very rusty. They have rusty scales, and it gives them kind of an orangish appearance, especially on the underside of the leaf. They also have these large terminal buds. There are a couple of plants that, when you're out in the woods, can look somewhat similar. Rhododendron canescens blooms around the same time, but its leaves are very different. It has kind of a palish, whitish underside, and they also have, the canescens has these very long curved stamens, whereas the chapmanii has much shorter stamens. So that's a big difference. And then the Leonia ferruginea, while it's in bloom, it's very easy to tell them apart. But they have very, very similar leaves. So this is what I mean by kind of an orangish, rusty color that shows up on the chapmanii. Okay, so rhododendron chapmanii wasn't always considered its own species. It was formerly considered a variety of rhododendron minus, and it was recently broken apart into a species of its own. So in this image, you can see the green area is the range of rhododendron minus, and the blue is chapmanii. So these 2 species occur in pretty different habitats. Minus stretches all the way up into the Southern Appalachians, where it occurs in kind of rocky escarpments. And in the Piedmont, it's also in rocky areas, kind of rocky cliffy areas or just rocky open areas. Down into the coastal plain, it occurs on these sandy bluffs. But rhododendron chapmanii occurs in mesic flatwoods for the most part. It can occur kind of in ecotonal areas between mesic flatwoods and depressional swamps. However, the easternmost population occurs in xeric hammocks, so a bit different than the majority of the range. Each of these circles, the larger circles that you see here, represents a population. So you'll see there are only 3 populations left. So this is a really important plant to do what we can to conserve while we can. This population, the easternmost one, is completely protected, and the southernmost one is partially protected. But this northern population is not protected at all. And unfortunately, it's also the largest population. The number one threat is habitat destruction, and that's the primary threat to that northern population. It occurs on private lands that have been converted to pasture lands and to timberlands. And so we don't know how much longer that population is going to be around. Overharvesting is another problem. It's a beautiful plant and people want to grow it. That can be done sustainably, but it could also be a problem if it gets out of hand. And then we all know too well that natural disasters are also a leading cause of species loss here in the Panhandle. So when a species is formally listed as an endangered species, the U.S. Fish and Wildlife Service, as I'm sure many of you know, comes up with a recovery plan and comes up with criteria that have to be met in order for that species to get delisted and go from endangered to threatened, or to eventually one day become secure. For this plant, the first one is that we want all 3 of those populations to be stable and to be protected. And unfortunately, we still have a lot of work to do in that area. The next one is that we're hoping to find 5 new populations, either discover a new population somewhere or create these new populations. And the Atlanta Botanical Garden is doing work on this, but this has not yet been met. This goal has not been met. And lastly, this kind of ties in with the first, we want to reduce the threats to these populations. So as this species stands right now, it is endangered. And so what is the Atlanta Botanical Garden trying to do to help it? First, I should say that we can't do this alone. We are funded by the U.S. Fish and Wildlife Service Coastal Program to do the work that we're doing. We have teamed up with FNA to conduct surveys, to look for new populations. And then we've gotten permission to go onto the lands where these plants occur from Florida parks. So it's really a team effort here. But the work that the Atlanta Botanical Garden is doing is a great way to highlight what ex situ work and in situ work really look like. So I'm going to be going through these different components with you now. So seed banking. Seed banking is a really important thing that botanical gardens can do for conservation, because we have the capacity to store seeds in such a way that they can remain viable for very long periods of time. And we have a seed bank at Atlanta Botanical Garden to do just that. So in 2020 and 2021, we collected over 3,000 seeds from Rhododendron chapmanii, and some of those went into our seed bank for long-term storage, and some of them got put into seed trials. So one of the questions with this species is that, in general, rhododendron tend to germinate very easily, but we're not really finding seedlings out in the wild anywhere. I don't actually know when the last time a seedling was found, and that was something that was included in the 5-year review of this plant, is that just seedlings are not occurring in the wild. So we thought we should look into the viability of these seeds. They're producing a lot of seeds, but are they viable? One way to do that is to use TZ scans, where you dye the seeds, and if they turn red when you dye them, they're considered viable, or a pinkish color too. And if they don't stain, they're not viable. So this image here shows a few examples. These kind of yellowish looking empty seeds are not viable. They just do not have an embryo. They cannot turn into a new plant. Whereas these darker red seeds are considered viable. So of the seeds that we put into these trials, 60% of them are viable, which seems pretty good. But when we then took it a step further and tried to see, okay, so the seed is technically viable, it could produce a new individual, but will it grow into a new individual? We had very different results. So we put some of our seeds into complete darkness, which a lot of seeds like to grow. And then some other seeds had 12 hours of sunlight and 12 hours of darkness. And we had very low germination rates. So we had a 16% germination rate for the plants or the seeds that were in the absolute darkness, and then only an 8% for the ones that had some light. So what this told us is that we have some more work to do in terms of finding out what conditions are necessary for these plants to germinate in the wild. Seeds seem to be viable at relatively high rates, but there's some condition that is missing for them to actually germinate in the field. But seeds aren't the only way that we can grow plants. So we have also been doing work with cuttings. So our conservation horticulture team takes cuttings that we take in the field and turns them into new plants. So myself and my field technician will go out into the field, find these plants, and look for the healthiest, greenest stem, making sure that we're not taking the only stem from a plant, of course. And we'll cut it and put it in a plastic bag with a moist paper towel, and then ship it overnight to Atlanta, where they will produce whole new plants, like you can see here. And these plants are ultimately taken out of their pots and planted at our conservation safeguarding nursery, which is in Gainesville, Georgia, so that they can be in the ground and really flourish. And then they provide material for future plants. So we have that plant there that we can take more cuttings from and grow if we decide to conduct an outplanting. But a caveat is that these are clones. So when we're taking cuttings, we are safeguarding an individual from the wild. So we know exactly who the mother plant is, and we know that the plants that we are growing are a backup for that mother plant. So if something should happen to the mother plant, we have a backup. But it's not a way to increase genetic diversity. So this is only one piece of the puzzle. We currently have 172 plants that we've grown from cuttings, and we're exploring ways to get those back out into the field, either in a new population or to augment an existing one. So this is something that I'm going to, I hope you don't have too many questions on, but if you do, I'll direct you to our research geneticist. But a genetic study is something that we really wanted to do with this species because there are only 3 populations, and we didn't really know how much genetic variability exists out in the wild. So what we did is the very beginning of a genetic study, you have to get tissue. So myself and my field technician go out in the field and collect leaves, and then we put those leaves on silica and then send them up to Atlanta where DNA is extracted, and then the study can take place. It's sequenced, and we can find out about the genetic variation within these populations. Our main question here was just how much variability is there? Are these populations actually distinct from each other, or are they all homogeneous genetically? We ideally wanted to look at all 3 populations, but the Camp Blanding populations, that farthest east population, it did not sequence well. That population, there is a little bit of a question whether it's even a wild population or whether it was planted. So that's something that we're still pursuing. But for the purposes of this study, we were only able to look at that southernmost population and then that northern unprotected population. Something that came up during this, though, is that the leaves that we collected from that northern population, we just didn't have enough. We had a really short amount of time to access that private property, and so we didn't quite have enough to do a rigorous study. So our scientists extracted DNA from herbarium specimens. And in so doing, they had enough samples that we could really look at this in a rigorous way. And what they found was that these 2 populations, so those 2 Western populations, they are genetically distinct. And why that's important is we have these clones of these plants, and we want to put those back out onto the land, but now we know we cannot mix them together. So if we take plants from that northern unprotected population, we don't want to move that material down into the southern population because we could create a loss of genetic variability. And some of that variability could be key to surviving drought or to surviving just extreme devastation from heat. So we now know that we have to take that into consideration when we're planning our future outplantings. The next thing that we did was a demography study. And I have a little diagram here. So essentially, our question with this is we looked at one population, that southern Gulf County population, and we wanted to know, is it stable, is it decreasing, or is it increasing? And so essentially what you see here is you have your population in the center, and here we have just some symbols for a really large, healthy, reproductive plant, kind of a medium plant, and then vegetative plants. So these are the different types of rhododendron chapmanii that we would find within a population. And then we have different factors leading to growth, and those are things like a flowering plant producing seed that creates a new individual, which again, I said we don't see much of in the wild. Or a different form of growth is a vegetative plant growing up into a reproductive plant. Or in clonal plants, like this one, you can see a vegetative plant resprouting, especially after fire. And that can be another way that a population could grow. And then the flip side of that is, what are the factors that are leading to decline? And those are mainly things related to death. So things like herbivory or drought or really, really intense fires. So pictured here is Ashlyn Smith. She was the former me. I took over for her after she accepted a full-time job with UF. And one of our field biologists, Caitlin Crocker. And they're out in the field. Here, you can see a lot of Chapman's rhododendron. You can see they're growing pretty dense together. That's a field biologist, Cami Adams. And then this is Ashton Holiday Goulart, both awesome people. And we really couldn't have done this study without them. So what you can see in that picture of how dense the rhododendron are is part of why we approached this the way we did. So like I said, we weren't really finding any seedlings in the field, and this is a clonal plant. So ideally, when you're doing a demography study, you want to follow one individual over time, but we can't really distinguish one genetic individual from another without a genetic study with this species. So we approached that by looking at stems instead. So we gave every single stem that was a part of our study its own numeric ID. And every year for 3 years, which was the amount of time we had funded for this study, we measured height of each stem, and we measured length of each stem. And those sound similar, but they're different because height is an unmanipulated measurement. It's just as tall as the stem is naturally, and length is stretching the stem out as far as you can stretch it. We also measured diameter, and then also reproductive characteristics like number of flowers, number of buds, and numbers of fruits. So Chapman's rhododendron, some would say needs fire, but we were hoping for the purposes of this study that fire would not be introduced to the stems so that we could kind of see how they all respond over a period of time where we were able to control the fire. However, fire was accidentally introduced to some of our plots, but not all of them. And at first that seemed like a really unfortunate thing, but it actually gave us the opportunity to investigate different burn intervals. So essentially what we ended up with was a nature-enforced experimental design where we had some stems that were burned within less than a year, and then others that were burned within 1.5 to 2 years, and so on. And so we were able to look at how these stems survived over time and what their reproduction was like. So one of our big questions with fire is, does fire change the reproductive status of the stems? And what we found is that the stems that were in that 0 to 1 year burn interval, they did not flower as much as any of the other stems, but none of the other stems were different from each other. So what that tells us is that burning these plants on a yearly basis would be too much, but anything more than that, and the stems, they really didn't differ from each other. So that was pretty good news for us. Okay, so here I have just a little snapshot of what our data looks like. So like I said, we gave each stem a unique numeric ID. Each stem had its own burn zone. And then these were the size measurements and reproductive measurements. So those are things we were actually measuring in the field. And then finally, we calculated other measurements, which were fate. Did the stem survive from year to year? And the stage that the stem was in, which is a size class. So unfortunately, plants can't just tell us what is relevant for them. They can't tell us what's biologically meaningful. They can't say, when I reach 20 centimeters, I am suddenly in a different size class than I was before. So we had to look at all of these different size measurements that we took and run some models to try to see which of those were most biologically meaningful. And what we found was that the length of the stems was most closely related to mortality. So shorter stems actually tended to survive kind of half and half. Medium stems, which we considered 23 to 100 centimeters, those had a much higher survival rate than any of the others. And kind of counterintuitively to me, the long stems were more likely to die. And any stems that were less than 23 centimeters were not reproductive ever. So we have all this information for every stem. And what we did is we ran some models in R using the PopBio package, which pretty much is taking every single stem's information and creating equations for each of these lines, like how often does a large stem create a new stem? How often does a pre-reproductive stem move on and grow up to be a medium stem or a large one? And from that, the package gives us something called lambda. And this is a number that is an approximation of how the population is doing. So a lambda of 1 means that the population is stable, and a lambda that's less than 1 means it's declining, and then a lambda of more than 1 means that the population is growing. So even though the stems that we were looking at are all within what we had thought was one population, we did split them apart into kind of pseudo populations based on their burn intervals, because they were just too different from each other to consider one whole group. So we did break them out into groups that were burned less often and groups that were burned more often. And what we found is that the stems that were burned more often had a lambda of 1.03. So they were growing, that population is growing. And the ones that were burned less often, so on, say, a 3-year interval, they had a very, very slightly declining population. I would take this with a grain of salt, though, because our study was 3 years long. That's not enough time to really, really investigate population trends. Furthermore, the only reproduction that we saw, we never saw seedlings. So we saw sprouts, but the sprouts were only a result of fire. So our approximation for population growth was those sprouts, but that was really only happening with fire. So of course, the model is going to kind of overestimate the importance of more frequent fire. So essentially, this did tell us some things. Yearly burns are too much. We're not going to get much reproduction if these plants are burned yearly. But we have a lot more questions. The graphs that you see here on the right show us what's called a stable stage structure. So it takes all that information that we collected and then projects over time. Okay, so those stems that were burned less often, if it continues, if those trends that we observed continued, how many of the stems would ultimately be in that pre-reproductive stage and how many would be large? In the stems that were burned less often, the majority of those stems are pre-reproductive. And there were also some medium stems, but there were almost no, according to the model, large stems over time. And we see something different in the stems that were burned less often. We see a lot higher frequency of large stems and some pre-reproductive ones. So those are questions that we have to ask ourselves. This model is looking at growth in terms of sprouts, but are large stems and medium stems more important than pre-reproductive ones? We don't know for this population. So we really need to continue more research, essentially. We also need to continue the question of why we aren't seeing seedlings in the field. what is happening where these seeds that are viable are just not growing, what conditions need to be created so that they can grow. How am I doing on time, by the way? What am I supposed to? Okay, cool. And then also another question is, when is the best time to burn? Because that could come into the reason that we're not seeing seedlings, is that maybe these plants need to be burned during the growing season, but they're primarily getting dormant season burns. So that's a question that more years of data collection could help us look at. Okay, I think that's all I have for Rhododendron chapmanii. I'd like to stop for a second. I did say I want to give a quick overview of some of our larger projects, but I was wondering if anyone has any questions about Rhododendron chapmanii before we move on. We can also let them come up later, but so that's a great question, and I often wonder that myself. So the way that Atlanta Botanical Garden wants to approach that is doing informed crosses, not in the wild, because once we mix the populations, we can't unmix them. So we could, however, mix them in a greenhouse and see how the mixed plants respond to drought, for instance. But essentially, if we did mix them, there's the potential that it would create more genetic variation, but there's also a potential that it would lead to homogenization. So some genes dominating and others eventually lost. There is not a ton of genetic variation within the populations. They are different from each other, but there's just not a ton of variation. So it would probably be pretty easy for that to be lost. But great question. I do think that that's something we definitely want to explore in a greenhouse setting first, and perhaps when it comes time to reintroduce populations, if we've mixed the genetics in such a way that we have these plants that are reproducing tons and their seeds are really fertile, then that would be something to explore further. Does anyone else have any questions before I move on? Go ahead. Good question. So I think that, and I guess I should repeat this for people online, just in case you didn't hear, if there's anybody there. The question is, are a lot of rhododendrons clonal? Is it common for rhododendrons to not reproduce via seeds? Is it common to not find seedlings for other species of rhododendron? I would say that a lot of rhododendrons are clonal, and when you see them, because of the way that they grow, because they are clonal and they become such dense networks, I think it is often hard for seedlings to grow because there isn't as much sunlight reaching the ground in, for instance, the Southern Appalachians where those rhododendron are growing. But seeds tend to, from our conservation horticulturist perspective, rhododendron seeds should grow really easily. And the low germination rates that we found in our seed trials is unusual. So they're generally considered a pretty easy to grow plant. So we're not quite sure what's going on with that just yet. Great question. No, and that's another question that we would love to explore. And that's a really important one because it's recommended that these plants receive fire, but we don't really know. And something else that I didn't mention is that easternmost population, the one that's occurring in xeric hammocks, it never has received fire. There are a lot fewer individuals there. There are only about between 30 and 50 individuals, but that population has never seen fire. But in general, it's understood and assumed that fire is very important for these plants, and it certainly seems to be in the Gulf County populations. But no, we don't know about that yet. Thanks. Are there any more rhododendron questions? Okay, I'm gonna quickly, do I have until 8 or should I stop before that? A little before? Okay, just stop me if I go too long. I just wanted to very briefly touch on a couple other big projects that we are part of. One of them is the Center for Plant Conservation's Florida Rare Plant Rescue. This is a project that's also happening as a different version, the California Rare Plant Rescue. So it's occurring in really biodiverse areas, and the goal is to back up all of the rare plants in Florida via seed banking. The plant pictured here is Asclepias ferridula, and that's one of the ones that we got to work with this year. These are all the partners in Florida working on the project, except for the Jones Center at Itchaway is another partner who is not on this list. They just joined the initiative last year. So here are some of the plants we've worked with so far this year. This is Hymenocallis henryi. This plant, the population that we were collecting seeds from was really badly impacted by Hurricane Michael. So you can see the habitat right here is really covered with debris, and it has not been burned since Michael. So I saw that and kind of assumed that there weren't going to be plants in there because these plants tend to grow along the edges of swamps. So I thought, that's probably a little too dense for them. But I crawled through, and I'm so glad I did because there were about 100 Hymenocallis henryi back there. Only a few of them were blooming, but we were able to take those seeds. So they are now safeguarded in Atlanta Botanical Garden Seed Bank, and we're trying to learn how to grow them from tissue cultures. And then this picture here is a wet prairie at the buffer preserve, a really, really beautiful wet prairie that we got to go and visit this year. A lot of plants are in wet prairies that are really important to conserve. We had been looking for one in particular for the last couple of years, Asclepias ferridula, and we did not find it. We were really disappointed. We had records of the plant being at this particular prairie, and it had been burned. This plant really needs fire. So this year we returned, and sure enough, there were hundreds. It was pretty amazing. It was really special to see. We did find a possible threat to the species. Somebody was munching all the flower heads off of these plants. So we had gone and counted and found hundreds, returned about a month later, and there were still flowers, but there were a lot that had been just completely munched off. So that is something that we need to look into as well. We don't know who was munching. Could have been deer, could have been some kind of insect, but we don't yet know. And another species that was growing side by side with that is this is Asclepias ferridula. This is Kufia aspera. That's another really fun plant to work with. They have really sticky flower heads, so the seeds were pretty difficult to collect. They just wanted to stick to your hands and stick to your clothes and not go into the bags that they're supposed to go in. This is a bog that we got to work in, working with Sarracenia rubra, subspecies golfensis, which actually has not been collected yet. We'll be going in a couple of weeks. And then Carex lutea is another one that was a huge success story. We found hundreds and hundreds of plants, all producing fruit. So we were able to collect a lot of seeds from that one. And our goal when we're collecting seeds is to collect from at least 30 different maternal lines so that we can have as much genetic representation as possible. And we want to collect from across the population. We want to collect from small individuals and really big, beautiful flowering individuals. So we really want to capture as much of that diversity as possible. So at those really large populations like the Asclepias ferridula and like the Carex lutea, we were actually able to do that. Sometimes you end up with only one seed, and that's all that you have. So the success stories are always very, very encouraging. Okay, really quickly, I'm just going to very, very, very quickly say we have a very cool project going on that gets talked about a lot. So I wanted to focus on a different story, which is the story of Chapman's rhododendron, but this one is very cool too. We are doing work at Deer Lake State Park over in Walton County to restore wetlands that have been degraded by really excessive tie growth. And we're doing this without herbicides. We're trying out different methods to see what works best to try to restore these prairies back to reference prairie conditions. I'm not going to go into all of that. But I will show you, this is what Deer Lake State Park looked like in 1941. For myself, as someone who is not from Florida, looking at this, it kind of looks like desolate to me. But the important thing, once you start learning about prairie systems and about grasslands, is that this is actually way more diverse than a lot of forests are. It's open, there aren't very many trees, there's a lot of sunlight, and there's high biodiversity. The next slide shows what Deer Lake looked like at the beginning of our project. To me, again, normally I would look at this and see green and think how beautiful, but all of the green almost in this image is tai-tai, which is native, but is not very great for wetlands because it really blocks out a lot of other plants. So just in a nutshell, in this project, we are removing the really heaviest areas of tai-tai mechanically, and so it is mechanical, it is destructive. However, we are seeing a lot of really cool plants coming back into these wetlands. We're reintroducing fire and we're having really good results. This should just sort of switch through some pictures. Okay, yes. These are pictures taken by Jeff Talbert, who is the project coordinator on a really large project happening at Deer Lake. I am the manager of a project funded by the EPA, and his project is a larger scale project funded by the Gulf Benefit Environmental Fund. So I just wanted to show you that so that if anybody wants to know more, feel free to reach out to me via email or afterwards. And if we have time for questions, I can take any now. These are all plants from the wetlands. Yes, in the back. Sorry, I didn't catch the question. So we are burning primarily in the not on 30A, but into the woods behind 30A, not on the beach side. I guess, well, Jeff is part of the burn team and he's on the fire council in his county. Maybe that's helped us. But we have a solid team of people out there, so it's always under control. Yeah, that's something else that in that before and after picture that I didn't point out is, yeah, in the 1940s, there were no housing development around there, but at the time of the project starting, there's a lot of homes right around the area. So that's definitely something that we have to plan around. Of course, planning prescribed burns, you always have to think about those things, but especially in those heavily populated areas. Awesome. Thanks, y'all. If you have any questions, feel free to reach out.